compete, and one of the groups is declared the winner. And it's an all-day kind of event, and there'll be about 300 people on campus, and they need all kinds of things. They need people to drive golf carts to get people around. They need people to proctor the exams. They have all sorts of stuff. So the more bodies we have helping, the better that will be. That's on a Saturday. Yeah, I don't think we would be able to do it on a weekday. So yeah. March 5th, Saturday, 1, 2, 3, 4 weeks from now. Is that right? No. 18th. So tomorrow's the 19th. Oh, I'm sorry, 2 weeks from now. 2 weeks from now. I was reading my calendar wrong. 2 weeks from tomorrow, technically. All right.
Is this open to all students or just like science students?
To help?
Yeah.
Oh, I think Dr. Reynolds would take whatever help she can get. If you have an English major friend who's a zombie, I'm guessing that Dr. Reynolds would take the help. Even if there was a penchant for brains.
Yeah, we just require like a certain amount of hours for.
She wants whoever helps to commit to 2 hours.
Yeah, and
you
then,
could.
Well, we need 3 hours at least a semester. Usually,
Oh, okay. Oh, yeah, sure. No, I mean you can ask Dr. Reynolds, but I don't think she would. She would balk. It's a colossal undertaking. I don't know why she does it. All right, so we're talking about predator-prey arms races, and we stopped at this slide because you guys hadn't read the chapter yet, so I asked you to read the chapter. The first part of the chapter, this is what they talk about. They talk about predator-prey arms races. They don't go into a ton of detail on predator-prey arms races, not nearly the amount of detail that you've studied predator-prey arms races in the past. So I want to kind of go through what's in the book, but then I want to remind you of what that predator-prey arms race was that we studied back when you were first years. So, what is the Red Queen hypothesis? And why is it descriptive of predator-prey arms races?
Isn't it like a species must like always like keep like evolving and adapting to the environment or situation?
So the scene in Alice in Wonderland is where it comes from, and Alice is in a race with the Red Queen, right? And what happens during that race?
I'm guessing she adapts.
Oh yeah, I did. Sorry. Yeah.
I'm guessing she does something. I don't know.
They're racing. As Alice speeds up, what does the Red Queen do? She speeds up, and then when she speeds up, what does Alice have to do? She speeds up, and then when she speeds up, Queen speeds up, speeds up, speeds up, speeds up, until they're both now going so fast, it appears like they're not moving at all, because as each one speeds up, the other speeds up to catch them. And so an arms race is like that in that because one species is selecting against the other, and then the other selects back on the one, they're both evolving in tandem, and as a result, neither of them ever really gets out ahead of the other, because as one advances evolutionarily, the other one responds with an evolutionary response. That evolutionary response acts back on the first species, which then responds evolutionarily. And so it's almost as if the two species relative to one another aren't going anywhere. And so this is the Red Queen hypothesis. Have you guys ever seen these people who experience weightlessness in airplanes? They do this thing. They call it the vomit wagon, I think. What they do is the plane. They do this to train astronauts. They have a plane and it climbs, And then at the top, it descends rapidly. And what happens is as you're climbing, you feel like you're being pushed back in your seat because gravity is acting on you. But then when you descend, all of a sudden, the plane is descending and you're descending, and you're all descending at the same rate. And because they do this turn quickly, when it first goes down, you're suspended in the air, but very quickly your rate of descent matches the rate of descent of the plane, and so you just stay hovering in the air. But both you and the plane are plummeting to Earth. But because you're both plummeting to Earth at the same rate, it appears that you're just hovering there in space. And then you get to the bottom, and then you experience the G's, you get slammed down to you grab onto the plane, and then it would slam you down to the floor, and then they make the climb again, and then they descend, and this whole time you feel weightless as the plane descends because the two of you are descending at the same rate, the plane and the person. But you're both plummeting downward, but because you're plummeting downward at the same rate, it makes it appear like you're just floating in midair. And so that's what's going on with the Red Queen hypothesis. This idea that species A exerts selection on species B evolves, species B exerts selection on species A evolves, which then causes it to exert selection on species B. This reciprocal selection goes back and forth. So, given that that's how predator-prey arms races operate, how do they begin? Do you guys remember what the life dinner principle is? The life dinner principle? Yeah. So in any given predator-prey interaction, if the prey loses the interaction, the predator-prey interaction, what does it lose? If the predator loses, what does it lose? It merely loses dinner. So who is under stronger selection generally? The prey, because the prey loses its life, whereas the predator only loses its dinner. So in order to have an arms race, if selection is heavier on prey than on the predator, what has to be different about a predator-prey interaction that leads to an arms race? Both of them have to lose their life in the process of losing the predator-prey interaction. So it becomes more of a life-life principle in a sense. So in order for a predator-prey arms race to begin, the prey has to be able to exert strong selection back on the predator. It's easy to see how the predator exerts strong selection on the prey, but the prey has to exert equally strong selection back on the predator. In the case of the newts and the snakes, the newt has a deadly toxin called Tetrodotoxin that has the potential to kill the snake. So the snake has a deadly effect on the newt, and the newt has a potentially deadly effect on the snake. Selection forces are then equalized, so that if the newt population gets a little bit more toxic, what does the snake population have to do in response? It has to evolve resistance, more resistance. When the snake evolves more resistance, what does that do to selection on the newt? Increased toxicity. That increased toxicity does what to the snake population? Selection acting for higher levels of resistance, and all of a sudden you're on your way to the races of a predator-prey arms race. What conditions are necessary for predator-prey arms races to continue? If this is a bad deal for the snake, why doesn't the snake just say, I'm not going to eat newts anymore? So maybe newts are the only things that occur there, but that's not true because they have frogs and slugs and other things that they can eat. Why? So in this case, there's a garter snake that preys on the newt. Why does that garter snake not just go and eat a frog or a slug or a worm instead? They might be more profitable, but they come at a potentially high cost. And they're decent sized newts. Do you remember anything about garter snakes from evolutionary ecology? I realize this is going back a number of years. There was a guy at University of Chicago at the time. Now I think he's retired at the University of, or he's retired at Oregon State. A guy named Steve Arnold, a geneticist at the University of Chicago. He was interested in the genetics of behavior, and he worked with garter snakes. And what you can do with a garter snake is you can take a cotton swab, a long cotton swab with a little ball of cotton on the end, and you can rub that on a potential prey item and then present the tip of that cotton swab to the snake. And what will happen with the snake is if the snake is interested in it, whenever you put something in front of a snake's nose, the first thing it does, it starts tongue flicking. And the tongue flick, the tongue is forked, it picks up scent particles and then puts them at the Jacobson's organ, this opening in the roof of the mouth that makes a connection to the nasal mucosa. So it picks up scent particles, then waves them around the Jacobson's organ, and that's how they smell. So when you see a snake tongue flicking, it's trying to get information about its environment through smell. And when you first stick a cotton swab in front of a snake, it's going to tongue flick. But if it's something that the snake is not interested in, it figures that out pretty quickly and it stops tongue flicking because that stimulus has no interest to the snake. If, on the other hand, it's something that the snake is interested in, the tongue flicking will continue and it will stay at a high rate for a period of time after you present the snake with the swab. And so they oftentimes respond to food scents as through increased rates of tongue flicking. And what Steve Arnold was doing is he was looking at the same species of snake out in California. But some of the snakes live on the coast and other snakes live inland. And those two snakes, when you look at what they eat different things. So one of them is like a slug and snail specialist, and the other one feeds on, I think it was feeding on worms or was it amphibians? But anyway, when he rubbed the swab on the slugs and snails and presented that to the snakes, the coastal snakes responded with a lot of tongue flicking and the inland ones did not. And when he rubbed it on whatever the inland snakes ate, and I don't remember what it was anymore, but when he did that, he would present it to the coastal ones and they wouldn't tongue flick at all, but the inland snakes tongue flicked a lot. When you took coastal snakes and inland snakes and bred them, crossbred them, what he found was that the hybrids behaved much more like the inland snakes, but they didn't behave any more like the coastal snakes. So he concluded that there was a genetic basis to this prey preference, although it was complex because it wasn't just the average of the two species. One of them kind of took over the other in the F1 hybrids. So there's this genetic predisposition to prefer a particular scent, probably because there's a genetic predisposition to prefer a particular prey species that you eat. And garter snakes are actually well known for this. Some garter snakes are frog specialists, some garter snakes are fish specialists, some garter snakes are slug and snail specialists, some are newt specialists. So, if you have evolved to specialize on newts, possibly because they're a good source of energy and also they're relatively abundant. If that newt starts to develop toxicity, why do you not just go and eat something else? Because I'm specialized. I eat newts. That's what I do. I don't eat frogs. I don't eat worms. I don't eat slugs. I eat newts. So now you have a population of snakes that have a prey specialization. We talked about this before when we talked about specialists versus generalists. You can choose to feed on one prey item versus another prey item and specialize on that because it's more profitable. But if that happens over a long enough period of time that it's always that prey item, you might develop mechanisms by which that specialization gets fixed in you genetically as a population. So now you're a specialist on newts, and now suddenly newts are a little bit toxic. If you don't have a genetic variation that makes you resistant, you go and eat a newt, and suddenly you're dead, whereas before you were alive. Some individuals in the population have the resistant variant, some don't. So over a couple of generations, you get rid of all the non-resistant snakes, so you're left with nothing but resistant snakes. They're not terribly resistant because the newts themselves are not terribly resistant. But now the snake is a little bit resistant, it can put selection onto the newt, and that newt population gets rid of all of the newts. The snakes get rid of all of the newts that are not terribly toxic, leaving only the most toxic newts behind. Now the newt population is more toxic, so when the snakes continue eating on the newts, any snake that doesn't have a resistance that is equal to or greater than the toxin of the newts, those individuals die, and now the snake population is more resistant. And it goes back and forth, back and forth. At any point, the snakes should just get out of the newt game. But they can't because they're evolutionarily canalized. They're evolutionary fixed to specialize on a particular prey item from an event that happened way before the newts got toxic. And that then ties them into that. So we tend to find predator-prey arms races in specialized predators that are so specialized that they can't just switch to an alternative prey. Because if they switch to an alternative prey, that does what to selection on the newt? Let's say these snakes said, well, those newts are too toxic, I'm going to start eating frogs. What does that do to selection on the newt? Hmm? They're not selected anymore, and as a result, they won't get any more toxic. And if it costs them some energy or something to create the toxin, selection will actually drive that toxicity out of the population. So in order for predator-prey arms races to continue, there has to be something that ties the predator into eating those prey. And so we tend to see these things only occurring in predatory specialists. If this goes on long enough, what happens? Just use the snake and the newt, for example. Thousand years of this selection going on. What are the newts like? What are the snakes like? Super toxic newts, super resistant snakes, because they're in this lockstep evolution. So this is how they begin, this is how they continue, this is the end result. But predator-prey arms races can come to an end. What might cause a predator-prey arms race to stop being an arms race? We already discussed one thing. The predator could just switch prey, and then that's the end, because then it's no longer involved with the other species. But there are other ways in which it can end. Genetic ways in which they can end.
If one of them like if the newts couldn't become any more toxic, or if the snakes couldn't become any more resistant.
Okay, so what you're basically saying there genetically is what?
They can't
Because they lack. I'm my most irritating when I ask you questions and refuse to give you the answer. And I know I'm at my most irritating when that happens.
I just feel like I'm irritating you because I don't know the answer.
Oh no, I'm not irritated. I am neutral. Hey, where is my stuff? Oh man, every time it does that. It is a different picture. I hope you're happy about that.
It shows your background and nothing else.
What's that?
So I think it's kind of odd that it shows your background. Like it's
Yeah.
showing you the food.
It's because it's mirroring. It's doing a split screen thing, but it wasn't doing a split screen thing a couple of minutes ago. Anyway, that's academic. So remember this from the theory of evolution via natural selection. This is heritability. Heritability is the correlation between the offspring phenotype and the parental phenotype. When selection acts on these individuals, it gets rid of all those parents, which means that we also get rid of all those offspring. The shift in the parental population is selection. This is the evolutionary response to selection, and that is mediated by heritability. But when you're done with that, all that is left is that cluster of points up there at the top. Whoa, up there at the top. Now, there's way less variation in parental height. There's way less variation in offspring height. And now look at what has happened to the heritability. The heritability was 0.96. Now it's 0.66. As selection acts on a trait, it reduces genetic variability. If we subjected this population to another episode of equal selection, it would get rid of this section, and that slope would be even flatter. And you keep selecting, keep selecting, keep selecting until eventually you just run out of genetic variation for the trait. The newts are as toxic as they can be, and the snakes are as resistant as they can be, and it just stops. There's no more genetic variation. Now, what are the odds that genetic variation is going to end for both species at exactly the same time? Not very likely. If the newt runs out of genetic variation first, what happens? Hmm? The snake wins, so it can the least resistant snake can eat the most toxic newt. And then what does the snake do? It eats all the newts. Then what happens? No more newts, and what does that mean for the snake? No more snakes because it's a prey specialist, isn't it? What happens if the snake runs out of genetic variation before the newt does? And then what happens to the newt? Woo! We won! We won! They have a party, and then there's no longer any selection on the newts. So then what happens to the newts? Well, they might, but what else will happen to them in terms of this trait of toxicity? Toxicity might, yeah, toxicity might decrease because there's no longer selection driving that, but it's only going to decrease if there's a cost to being toxic. Sorry, I have something in my throat. I was in a meeting today after statistics. It's supposed to last 20 minutes. It was an hour and 40 minutes. And I was in charge of delivering the material in the meeting, so I've been talking a lot today. Mainly because there were a lot of questions. So the newts probably become less toxic because there probably is some cost to producing that toxin. And in the absence of selection to make them more toxic, that cost then imposes a selection to make them less toxic. But in the end, the snake goes bye-bye and the newts are still around. Yay, celebration for the newts. So that's how that's how they can end. One or the other species runs out of genetic variation. Depending on the species, that spells different outcomes, but the root cause is a lack of genetic variation. Do they end because the predator just stops being a prey specialist? Does the predator-prey interaction end because the predator stops being a prey specialist? Yeah, probably doesn't. Plus, it's really easy to evolve out of prey specialization after you've evolved into it. The act of becoming a specialist shuts off lots of other avenues for you. In the case of snakes, their brain has become attuned over long periods of evolutionary time to pay attention to only certain types of scent cues. To evolve them out of that is very difficult because if it was easy, what would have happened at the beginning of the predator-prey arms race? Yeah, they would have evolved out of specialization into generality a long time ago. Is there another evolutionary process that we will study in animal behavior that is analogous to a predator-prey arms race? Hmm? There is. There definitely is. Anyone? Bueller? Oh, it did it again. Some. And where's my cursor? Is there a cursor up there? There is a cursor up there. Okay, so it went all the way over there. It's going all the way over here. But it doesn't ever appear on my screen. That's just ridiculous. Ah, there it is. No, not Peacock TV. I don't want NBC. I want an actual Peacock. Ah, Peacock. What do you notice about peacocks other than that they're ridiculous and loud? They have pretty tails. They have also long tails. This is a widow bird. What do you notice about widow birds? They have long tails. I don't know how loud they are, but they're just black. But they do have nice red and yellow orange, red and yellow wing patches. Um, what do you notice about barn swallows? It's long. It's longer than other swallow species relative to their body length. And it's mainly males. Males have much longer tail feathers than females do. So in all three of these cases, the males have really long, elaborate tail feathers. Why do these bird species have really long, elaborate tail feathers? So let's look at this figure here.
Mating.
Oh yeah, this is a male, this is a female, this is a male, this is a female, this is a male, this is a female, three different species. This is the barn swallow, and you can see that the males are longer than the female. Sorry, what was that you said, Avery?
And like some type of like mate attraction.
Oh yeah, long tail means sexy male. So in these systems, the females are choosing the males to mate with or not. And the main thing that they are cluing in on is tail length. You have a long tail, you sexy male, I want to mate with you. If there's a genetic basis to tail length, then selection can act on tail length such that the more the females choose long-tailed males, the longer the tail gets on the male. But you can also have a genetic basis for choice in the female. If females that are choosier about the males that they want to mate with, they end up producing sons that have longer males, which means that they get more females. So in this way, male tail length becomes genetically correlated with female choosiness. And as soon as that happens within a species, females exert selection on males for longer tail. Being longer tail then makes it possible for selection to act on female choosiness, such that females who are choosiest mate with the longest tailed males and have more offspring than if they chose to mate with a less long tailed male. And so these two things get coupled with one another. Male tail length gets longer and longer, and females get choosier and choosier. What was a sexy long tail 10 generations ago is just now a ho-hum long tail now, because female choice has also evolved. So sexual selection is one of these situations where you can be in an arms race, but in this case, it's an arms race not between resistance and toxicity. It's a arms race between the male phenotype and the female preference for that male phenotype. Does that make sense? Does that resonate with you?
Isn't that kind of like what the kids did? Didn't they choose like the offspring like would that happen somewhere?
So yeah, you could envision it happening there, such that the babies might get more and more brightly colored, and as a result, the parents' preference for feeding the babies might evolve. Sure, that could work, because once again, it's a preference thing. The dynamics are a little different where preference is concerned, because the consequences aren't nearly extreme, so we see evolution oftentimes happening slower in those situations because it isn't a life or death thing. It's more of a reproduction or not reproducing thing. And oftentimes it's not even reproducing or not, it's reproducing more or less. And so just overall rates of selection aren't as high because the consequence of having a slightly shorter tail aren't nearly as much as dying. If you have a slightly shorter tail, you may only mate with 2 females instead of 5 females. You're still mating. You're still getting it out there. I just said getting it out there in a class.
And it's recorded.
What's that?
I said, and it's recorded.
And it's recorded. Oh my gosh. Oh my. Something happened to the recording. It didn't get posted. Okay. What's that?
Sorry, Jason, I normally
Oh yeah, too bad.
come to class.
Poll was inappropriate on tape, so I had to delete the recording. No. Um, so Davies et al. Makes the authors of your textbook make this distinction between really talking about adaptation as a process versus a kind of storytelling. And Stephen Jay Gould was the first person to try and make this point. Um, an adaptation I like to think about a lot is the thumb. We as humans have opposable thumbs. Now, I could tell you a story about hitchhiking, and I can craft that story so that you would believe that selection in the 1950s and 60s, acting on people who needed to move around a lot but didn't have any money, favored people who could use an opposable thumb for hitchhiking, because if you were here, your girlfriend was in the town next door, and you didn't have a car, you could hitchhike to go and hook up with your girlfriend, and then you would procreate, and then your genes would get into the next generation. And that's a ridiculous story because we know why the thumb evolved. The thumb didn't evolve that way. But that is a plausible story. People do use their thumb for hitchhiking, and hitchhiking can, in some cases, increase your reproductive success. But that's a story that you create that can account for the thumb. Can you tell other stories about the adaptive function of the thumb? Stories that are probably not true. Can you think of any? That's because you guys are probably way too fact-based for this particular approach to teaching here. Did I bring my phone? I didn't bring my phone because I don't like my phone going off in class. I'm on Tinder. Oh, oh, who's that? Swipe? Oh, swipe back. Is that how Tinder works? Do most people have to swipe on Tinder? Most people have to swipe on Tinder. Okay. She swiped back. Oh, oh, she's sending me a text. Got a text. Oh, I'm texting back. Text to me, text back to her, text to me, text back to her. Want to get together tonight? Sure. One thing leads to another, and now you're married and have babies. Thumbs evolved because of selection, acting on our ability to text, because that's how you hook up in the year 2022. And being able to do that effectively might influence your reproductive output. Is that why thumbs really evolved? No, it's ridiculous. But it's a story you can tell, and it's plausible because people do use their thumbs for texting. But the problem with all those things is that they're just a story that you can tell. They're not really, thumbs are not really an adaptation for texting in the same way that thumbs are not an adaptation for hitchhiking, which actually can have negative effects on your fitness in reality when you get picked up by an axe murderer. What is probably the real adaptive function of the thumb? For grasping things, right? I wish I had brought a stick with me. And I don't have anything stick-like. Wrap your fingers around a stick without hooking your thumb there, and then ask a friend of yours to try and rip that stick out of your grasp. And they're going to be able to do it pretty easily. But as soon as you hook that thumb around, it becomes way harder to take that stick away from you. Now imagine that we are early primates and we live in trees. Having an opposable thumb is super useful because you need a strong grip. And so you can see digits that were normally all the same direction becoming opposed because selection might favor individuals with a stronger grip. So over evolutionary time, that becomes what the norm is for primates. So that the thumb is an adaptation for grasping things firmly. It is not an adaptation for all these other things that we could tell stories about. And we could actually test that by looking at strength of opposable thumb species versus grasping strength of non-opposable thumb species. We could look at the early ancestors of the primates and ask ourselves, did they have opposable thumbs or did they have non-opposable thumbs? And then we could ask ourselves, based on what we know of their diet from fossil evidence or whatever else, what kind of environment did they live in? Were they living in trees or were they down on the ground? if they were down on the ground and they had non-opposable thumbs, and then the earliest primates that we have that were living in trees had opposable thumbs, that's a comparative method way of thinking about the adaptive function of the thumb. You only see the thumb in those groups that ended up being in trees, etc. So adaptation is a very specific thing. It's a trait that increases your fitness that evolved for that function, whatever that function is. The thumb is not an adaptation for texting because the thumb might increase your fitness, but it didn't evolve for that particular purpose because when the opposable thumb evolved, cell phones weren't a thing, needless to say. So making this distinction is important. Is snake resistance an adaptation to newt toxicity? Some people, like Stephen Jay Gould, would argue only if that resistance came about in response to selection to newt toxicity. In this case, because it's a very tight interaction, one species of newt, one species of snake, it's probably a pretty good idea that those two traits, toxicity in newts and resistance in snakes, are adaptations designed to, in one case, thwart the snake, and in the other case, to eat the newt. Questions? All right. So this is a series of papers that you've already read in your distant past. First of these published in 1990. Edmund Brody III is Butch Brody Jr.'s son. E. Brody Jr. was department chair at the department where I was a PhD student. I graduated, no, I didn't go there yet. I entered the graduate program in 1991. So father and son team. Back in the 1960s, Edmund Brody Jr. had looked at newt toxicity because he went to his professor at the school where he was an undergraduate. He wanted to do undergraduate research at a time when undergraduate research was really, really rare in the 1960s. And said, hey, I want to do a research project. And his professor said, oh, well, there's this news report about these fishermen who they found dead out at their fishing camp, and they don't know how they died. But the only thing that they suspect might have happened is they might have been poisoned because when they looked inside the coffee pot, they had brewed themselves some coffee that morning, and there was a newt that was boiled in the coffee pot. Do you remember the story from first year?
No, I'm just surprised there was a newt in the coffee pot.
Well, they went down to the stream, scooped up some water, didn't pay attention, didn't look in the thing, scooped up water, put the lid on, put it on the fire, poured the coffee in, put the lid back on, and then drank the coffee.
How big is a newt?
They can be reasonably long with a tail and all, but it doesn't matter. If you're in a population where they are really, really toxic, one newt can easily, evidently, kill 3 men. What? What?
In your coffee pot?
In your coffee pot?
You didn't notice that?
Well, I mean,
No, but like,
no, metal coffee pot, you scoop it up, you put the lid on, you carry it back. So, I can't believe I'm having to argue the mechanics of coffee pot. You're in your tent. You get out, pull on your suspenders. The stream is over there. You're half asleep anyway because, of course, you haven't had any coffee. You go over to the stream. You squat down. You scoop up some water. You put the lid back on. Why are you putting the lid back on? So you don't spill the water when you come back. You set it on the grate on the fire. You wait for it to heat up. Then you open up the pot, pour in your coffee, put the lid back on the pot. I can easily see how you would get a newt in your coffee pot and not notice it. Not me, because I don't drink coffee. You know why? Because you drink coffee and the newt kills you.
maybe that's why they make blue coffee pots.
What's that?
I said maybe that's why they made clear coffee pots.
You probably wouldn't have a clear coffee pot on your camping trip anyway.
Oh,
Probably have a metal one. I don't eat almonds either. You know why? Almonds smell like cyanide. And you don't know if you're just eating an almond or if somebody's trying to poison you.
Hmm.
Coffee and almonds, stay away from them because they mean certain death, maybe. So it killed these three fishermen. One newt killed these three fishermen. So his professor was like, see if they actually died from the newt. So he went off and he got the newt and fed pieces of newt skin he fed pieces of newt to a wide variety of different predators and found that it killed most of them. But he fed it to garter snakes, and lo and behold, some species of garter snakes would eat the newt and they would be fine. Some cases they would eat the newt and they would be knocked out for a little while, but then they would come back to life because they weren't ever actually dead. They were just immobilized. So he wrote a little paper on this and then just didn't really do anything with it from then on, but it set him on a path of thinking about the skin secretions of amphibians and what their properties were in an anti-predator sort of way. And so he had been doing work on Asian salamanders, et cetera, et cetera. And then his son, Edmund Brody III, was back at his grandmother's place gathering snakes for his doctoral dissertation in his grandmother's backyard in Oregon. And whenever he would find a snake, he would palp its stomach to see what it had been eating. And he was surprised to find that some snake species, Thamnophis totalis in particular, he would occasionally palp up a newt, get it to regurgitate a newt. And he was surprised at that because he had learned in the past that these newts were toxic, and he was surprised to see perfectly functioning snakes with a newt in their gut. And so he mentioned this to his dad, and he's like, well, of course you know that they're toxic because your dad, found this out back in 1968, and I've told you the story that snakes are resistant. Don't you remember anything I tell you? And so they started talking about it, and they were like, well, let's explore this more. And so what they did was they went back to Oregon, and they collected Thamnophis totalis in Oregon, and they collected Thamnophis ornoides, which is a different species, but it's a slug specialist. It eats gastropods and worms. It lives in the same place that Thamnophis totalis lives, but Thamnophis totalis eats newts. Thamnophis ornoides doesn't eat newts. And then there's a population of Thamnophis totalis in Idaho that can't eat newts because newts don't occur in Idaho. And what they did was they injected them with different levels of tetrodotoxin, from hardly any tetrodotoxin to a lot of tetrodotoxin, and then they looked at how fast their speed changed. So they would race the snakes down a racetrack, record their speed, then they would inject them with tetrodotoxin, and then race them down the racetrack again and see how fast they were relative to their initial speed. And what you see is that the more tetrodotoxin you give to a snake, the slower it becomes, because presumably the tetrodotoxin is interfering with the locomotor ability of the snake. And the Thamnophis totalis in Oregon that live with the newts and eat the newts was affected least. It took a lot more tetrodotoxin to knock them down than either the Thamnophis ornoides that lives with the newt but doesn't eat it, or the Thamnophis totalis that would potentially eat the newt but can't because it doesn't occur in Idaho. Those were both very similar in their resistance to tetrodotoxin. From this, they concluded that there is an evolutionary response of the snake to toxin that is causing selection on the snakes as it's produced by the newt. And they found that this resistance was heritable. So heritability was actually quite high, 0.715, and that's a pretty high heritability value. So snakes can evolve resistance. It turns out tetrodotoxin is a compound that blocks sodium channels in nerves, and so it paralyzes the individual. It can paralyze you to the point that it actually stops your heart from beating, stops your breathing, and can kill you. And so it acts on a particular type of cell in a particular way. It prevents sodium ions going through the sodium channel, so you can't propagate a nerve impulse. And that's crucially important because that affects all your muscles in your body. So then they wanted to know, kind of, is there a cost to this? So they looked at a couple of the populations that they had looked at before. Benton County was the first. This Stemnophis totalis in Oregon, this is in Benton County. And so they looked at in Benton County, they looked at 10 Mile, they looked at Vancouver Island. In these cases, these are all places where the newts presumably vary in toxicity because the snakes also vary in resistance. Benton and 10 Mile are two places where the snakes are pretty resistant and the newts are pretty toxic. Vancouver Island, not very resistant, not very toxic. And what they found was that the dotted line is the one-to-one line. So if we were to put you on a racetrack down at the track here at Williamsville College, we would test your baseline speed from slow to fast, and we would measure your post-injection speed if we injected you with tetrodotoxin, which we are not going to do, by the way. If you, from slow to fast once again, some of us are slow and some of us are fast. But if tetrodotoxin has no effect on you, what would you expect the effect of speed to be? It'd be the same. If you started out slow, you would be slow after being injected. If you started out fast, you would be fast after being injected. This should be a one-to-one line. Slope equals 1, right? Because you would be the same speed after being injected than before being injected. If, on the other hand, tetrodotoxin has a negative effect on you, what would you expect to happen? Would it be a decreased slope? Would it be a change in slope, or would it be a change in y-intercept? It would be a change in y-intercept. Everybody would go down, but if everybody goes down the same amount, the slope doesn't change, it's just lower down. Everybody's slower, and everybody's slower the same. But that's not the case because the dotted line is the one-to-one line. This is actually the best fit line for the data. The best fit line for the data shows that those individuals that are fastest in their baseline speed slow down more than those individuals that are slowest, which slow down less after they've been injected, which then results in a change in slope. So now the slope is less than 1. The y-intercept is also lower, but the slope is less than 1 because these are affected more than these are in terms of their total speed. The next figure illustrates this. Whoa. This is the relationship if there's no effect of tetrodotoxin, and there's also no trade-off. If you are 5 centimeters per second baseline speed, you're 5 centimeters post-injection speed. If tetrodotoxin has an effect on you, but it's the same for everybody, you should just expect this line to go down. So when you actually go out and test this, you'll get a set of data, and when you find the average value of baseline speed and the average value of post-injection speed, you can move that one-to-one line down to where it intersects the average. And that's what you should expect if there's no effect of tetrodotoxin and no trade-off. But what we actually find when we fit a best fit line through the data is we find that the regression line is this. And what it means, once again, is that fast snakes are affected way more by tetrodotoxin than slow snakes are. They're all affected, but fast snakes decline in post-injection speed much more than slow snakes do. So there's this trade-off between baseline speed, how fast you are when not challenged by tetrodotoxin, and the effect that tetrodotoxin has on you. If you are really fast, tetrodotoxin affects you a lot. If you're normally pretty slow, it doesn't affect you much at all. The reason for this is probably physiological. If you're fast, it's probably because you have very good functioning sodium channels. So any little messing with those sodium channels messes them up such that it really slows you down. If you're slow already, you probably have sodium channels that don't function that well to begin with. So tetrodotoxin coming along and messing with the function of them doesn't really change things that much for you as an individual. And that's kind of a physiological explanation for it. There's a much better biochemical explanation for it, which I'll get to in a little while. But as it turns out, because the solid line, which is the actual data, is always lower in slope than the dotted line, which is the no trade-off, there's a trade-off between sprint speed and resistance, because the fastest snakes, baseline speed, are the least resistant snakes. The slowest snakes, baseline speed, are the most resistant snakes. And that's the nature of a trade-off. We've talked about trade-offs in this class already, where as one thing increases, the other thing decreases. As your sprint speed increases, your resistance level goes down. And that's what they found here. So in later papers, what they did was they looked at actual variation in the sodium channel. So the sodium channel is a structure. It is made up of 4 subunits. Each subunit is coded by a different domain of a gene. Domain 1, domain 2, domain 3, and domain 4. These things form essentially a pore in the membrane. So these are transmembrane proteins, and they're bridged together by these linkers. So domain 1, domain 2, domain 3, and domain 4 are bridged together by these linkers. And these linkers are on the outside of the cell. So this is extracellular up here and intracellular up here. And it turns out that there is a series of mutations that occur in the DNA that code for amino acids in this particular linker right here, domain 4 on the outside of the cell. And it turns out that some of these are more important for causing resistance than others. So when we look at for some reason my pointer just wants to be absent today. When we look at this particular amino acid, it is a change from aspartic acid to asparagine. D is aspartic acid and N is asparagine. And in a couple of places where we see this, we see that those snakes are by far the most resistant, 100 times more resistant than the next most, 100 times more resistant than a non-resistant snake. So there's something about that particular mutation in this section of the sodium channel that makes these snakes very, very resistant. Changes in the other 3 domains of the sodium channel don't seem to matter. And so when they took these sodium channels and cloned them and put them in frog eggs, you can actually expose them to various levels of tetrodotoxin, and you can see how well they propagate nerve impulses. And so you can see the proportion of unblocked to blocked sodium channels is here at the 50% point. And we have these 4 populations here. We have an out group from Illinois But then we have Bear Lake, which is not very resistant at all. Warrenton is more resistant, and Benton County is more resistant, and Willow Creek is the most resistant. And what we see when we look at the effect of tetrodotoxin on sodium channels that have been isolated by basically taking the gene for sodium channel and putting it into a frog egg, so that the frog egg basically produces the altered sodium channel, the mutated sodium channel, they occur kind of in the order that you expect. Least resistant, next most resistant, super resistant at Warrenton. So it appears that tetrodotoxin is actually functioning through the sodium channel, affecting the ability of a sodium channel to propagate a nerve impulse. This is the propagation of an impulse. This is for the least resistant population. This is without tetrodotoxin. This is with tetrodotoxin. And you see that nerve impulse doesn't get propagated. This is for Warrenton. You can't see it very well, but there's a red line on top of a black line. The black line is without tetrodotoxin. The red line is with tetrodotoxin. If we blow that up, you would see that there's black right there. If you have the original paper, you can blow it up and you can see that there are two lines there. They're just right on top of one another. Not affected by tetrodotoxin hardly at all. So they did that with these 4 populations, but then they expanded and they wanted to look at the whole geographic range of where these 2 species overlap with one another. And what they found was that prey toxicity varies quite a lot, and predator resistance varies a lot. But if you put these 2 maps on top of one another, you can plot where there's a phenotypic mismatch, where either newts are really toxic and snakes are not very resistant, or a mismatch where newts are not very toxic and snakes are really resistant. And what they found is that there are some areas where there are significant mismatches, and they appear in purple here. These purple areas are areas where the newt and the snake have phenotypes that are not matched. But when we plot these in terms of predator resistance on the x-axis and prey toxicity on the y-axis, what we see is that in this area there are a number of populations. There are no populations here, and there are some populations over here. This is where the predator-prey arms race is presumably going strong, because the newt toxicity is similarly matched to the snake phenotype, which means that as the newt changes, the snake changes. As the snake changes, the newt changes, because they're kind of equally matched. This represents an area where the snakes are totally incapacitated by the newts. This area represents an area where the snakes are resistant and are unaffected by the newts. So in this case, the newts are really toxic and the snakes are not very resistant. In this case, the snakes are really resistant and the newts are not very toxic. In every one of the cases where there's a mismatch, it's always the snake is very resistant and the newt is not toxic relative to the resistance of the snake. Now those newts up there in the upper right-hand corner, those are still pretty toxic newts, but the snakes are way more resistant than the newts are toxic. These ones down here are probably situations where the predator-prey arms race hasn't started, because neither the snakes nor the newts are very resistant or toxic. So in all cases, one of these species is winning the race, and it's always the snake. So we talked about ways in which the predator-prey arms race can end. We talked about species running out of genetic variation, but there's another way that the predator-prey arms race can end, and that is by one species evolving much faster than the other, so that one wins the race and the other one can't keep up. That appears to be what's happening with the snakes. They have evolved high levels of resistance much faster than the newts have been able to evolve toxicity. So how can this happen? We will talk about that on Monday. Questions? All right, I will see you on Monday. Have a good weekend. Try and stay warm. It's supposed to be warmer.
It's
So,
supposed to snow Wednesday or Thursday again.
what's that? Yeah, and then it's going to get warmer and then it's going to get colder because that's what we do here in Missouri, I guess.
It was almost that Thursday.
Yeah.
We shot it out so far.
Should have put my lab on Tuesday, I guess. Alrighty. I have to go meet with a bunch of applied stats students. Yeah.